after the early session, I changed a few slides. I will just focus on the neonatal heart regeneration cell cycle and bioinformatics. So the title is the remuscularization of injured ventricle from within and outside in large mammal. So I will not talk about the transplantation of the cell and cell products and the engineered tissue because it's limited 14 minutes. So I will just focus on the, following the earlier session, talk about the P1, P7 or neonatal heart regeneration. What is the regulator? So this is an old slide. My lab has done thousands of dog heart. So this is old, actually this is a pig heart, arrow points the thin piece of scar, second to myocardial infarction. This is the LV dilatation that go almost 30 years ago. We are still trying to work out to remuscularize that piece of the thin scar secondary to myocardial infarction that happened 4 weeks before this cardiac MRI was taken. So remuscularization of injured ventricle has been achieved, as shown here. This is using the human iPSC-derived MHC-driven cardiomyocyte, or the MHC-driven D2 overexpression. So only the myocyte differentiate to, only the myocyte differentiate from the iPSC then start overexpress of D2. When injured heart receive these human iPSC-derived MHC-driven myocyte in the neonatal, in the immunodeficient mice. So 4 weeks later, the graft MHC-driven cardiomyocyte continue to proliferate. So it's a significant piece of a graft in the immunodeficient rodent heart. And 6 months later, the transplanted grafted hearts continue to divide. So it have a significant of remuscularization in the rodent heart with myocardial infarction, as shown here in the green is the confocal of red human-specific protein and cardiac. This shows that the significant graft of a human iPSC-derived MHC-driven myocyte in the rodent heart 6 months after the transplantation. So this is in the rodent. I will focus on using the neonatal pig model to try to examine using the strategy to turn back the clock of myocyte cell cycle to remuscularize injured ventricle. So this is my collaborators in UT Southwestern Hashan CDEX lab showing in Science about 10 years ago that rodent, just like a fish, can remuscularize injured ventricle, but it happens only at the P1. So at P1, you apex resection by the postnatal day 26, they grow completely grow the apex. So by then it was shocking our field. So we were first time learned that the mammal actually can remuscularize a significant piece of heart, but it only happened in the postnatal day 1. So after that, in 2018, this is the 2 reports from 2 continents. One is from the UAB, my lab, and then another one from the Singapore National Heart Institute. At the same time, report that the large mammal, specifically in pig, can also regenerate injured ventricle, as shown here, that the AOD ligation in postnatal day 1 by the postnatal 4 weeks later, the apex, injured apex is replaced by the regenerative muscle. So then one of the trainees, like one of you that in the lab, was thinking, if 4 weeks after the apex recession or apex injury in this particular study, if the 4 weeks later there's a regeneration of ventricle, then there must be a machinery continuous generating these myocardia. So that's you call rejuvenated heart, or it's regenerative heart. So in that system, if we do another second injury, AOD ligation, whether the heart can repair itself. So therefore, we did this study. In this particular study, we did 100 pigs, and then following that study, we continued at the neonatal pig model, try to remuscularize injured myocardial infarction heart beyond the postnatal day 7. So if we can make the adult heart remuscularization in the large mammal, that will be more impactful for the public health. So then we did another 100 pigs. So these are published in Circulation. So this one published 2 years ago. This second one is about the single nuclear RNA-seq analysis of cardiac cell in the neonatal pig, just published 4 days ago in Circulation. So now all the single cell RNA-seq data are on the cloud. So the Circulation requires to upload all the data. Data upload takes 3 days. So these data are available for every one of you if you are interested in normal heart. For example, this study we've done about 50 pigs. So we have the normal heart, fetal heart E80, that is 30 days P minus. That is P minus 30. So we have a fetal heart, single nuclear RNA-seq, all the raw data in the cloud. Then we have age-matched normal. These are naive heart get into the lab at the postnatal day 1, day 28, and day 56. All the raw data in the cloud. And then non-regenerative heart, that is the normal heart, at the P28 have AOD ligation, and then we examine 4 weeks later at P56. So those are the classical AOD ligation. We will have the scar. And finally, this is the study group. We examine them. That's the diamond of the study. That's the regenerative heart. We call regenerative heart. Have a double injury. So P1 apex resection, and P28 AOD ligation, and then we follow up 4 more weeks explant heart at P56. And then subgroup of animals, 2 days, 7 days, 14 days after the AOD ligation, we explant heart to see early phase after AOD ligation of this double injury model if there's a scar. So now, this is the single nuclear RNA sequence data, the all raw data in the cloud. Minnesota has access, and UCL has access, and any one of you want to analyze the normal heart from P minus 30 to the adult, all data are there. So, and I would just going to only 14 minutes, I only can show you a little bit of the raw data and the finding, very preliminary. So this is the typical of the heart at P50 that experienced double injury. So apex resection, it healed. This is looking from inside out. Look at the anterior wall. We did not find any scar. And angiography showing the AOD occlusion distal to second diagonal is successful. This is before explant heart. We did the angiography in showing the AOD occlusion. And then we look very carefully from longitudinal look at the LV following the coronary anatomy. So AOD perfused by the myocardium perfused by AOD are the septal wall, anterior pulmonary muscle, anterior wall, and this is the remote area perfused by LCX. So there's no scar 4 weeks after the AOD ligation in double injury model. And typical 2 days and 7 days after the AOD ligation, those are the double injury model. Again, it's the P1 apex resection, P28 AOD ligation. 2 days after the AOD ligation or 7 days after AOD ligation, we did a TTC. There's a clear scar, significant scar. That's 60 minutes complete AOD occlusion and re-established flow after 60 minutes of occlusion. So these are, therefore, there's definitely early after AOD ligation in double injury model, there is scar. And this shows the, by week 4, P56, 4 weeks after the AOD ligation, regenerative heart can completely regenerate the AOD perfused area as shown here. This we cannot find by naked eye any scar, and the non-regenerative heart, meaning they've not experienced the apex resection at P1. When you do an AOD ligation, you see a big piece of yellow scar as shown here. Just by naked eye, you can see a thin piece of scar. My lab have done thousands of pigs, so this is very consistent, large piece of scar. So what is going on? That's the information, the cell cycle information. So this is the indication of the cell cycle by Ki-67. So the P1 normal heart is at this level and decreased to close to 0 at the P28, P56. And the ones with P1 injury, the Ki-67 positive versus total nuclear is even higher than normal heart. So it's really the P1 injury make the entire ventricle at the regenerative state. This is it's not only the apex area, it's remote area. It's surprisingly also upregulated the cell cycle activity. So likewise, this is another cell marker, cell activity marker, PH3 positive NAK 2.5 positive cells in the normal heart, newborn at the P1 is at this level, and the P28 decreased to almost 0. And at P56 normal heart is 0, but the regenerative heart in the large mammal, when they experience the P1 apex resection, the cell activity level remain high as shown here. And the remote is, for whatever reason, the remote area that perfused by LCX, the cell activity is remain high too, as shown here. Similarly, the Aurora B, symmetric Aurora B as shown here, normal heart P1 is at this level, decreased to 0. at the P28 and P56, but maintain high in the double injury model at the P28 and P56. And typical Aurora B, symmetric Aurora B is shown here. Now it's the single cell RNA-seq neonatal large mammal heart. So now all these raw data are in the cloud. So again, the raw single cell nuclear, lots of data. It's taken 3 days to upload. It's completed. So it's UCL using it. I would be happy to have Oxford or Australia using these data also. So we have now looked at the macrophage, for example, the subpopulation of the red one here, pink one here. It's like a thing of the goldfish. These are data available for people interested in the macrophage. We are only interested at this point only the myocyte. So these are the population of subpopulation of myocyte. And let's look at the Leuven clustering identify 6 cardiac myocyte clusters as shown here. So in particular, there's cluster 6 is the fetal heart. It's all here. And cluster 5 is from this, it's PCAM, we call PCAM2 activated heart. It's a complete different subpopulation. And all these injured heart are CM3. So it's the CM3 is the fetal P1 heart, and all the injury are in here. It's the CM1. So again, the CM5 is the PCAM2 positive. So this is the plot, trajectory plot of apex injury followed by P28 AOD ligation. We see the injured AOD myocyte goes to CM3. That is the P1. It's a trajectory plot. So it goes to fetal, go to P1, goes to the fetal heart, and then goes to the PCAM2. So that's the warning, right?
The window opens
so I'll
automatically
summarize,
after 10
finish
minutes.
here. I can read it, no problem. So in summary, so this is the lots of data, but we make this summary for now. We have established a large mammal neonatal model with the double injury and established for the first time to prolong the window of remuscularization to P28. So that's important conclusion. And then secondly, we have generated single nuclear transcriptome of cardiac cells from neonatal, from actually fetal to neonatal to adult, and then injured pig hearts. And that's the raw data in the cloud. And this summary, we talk about ongoing work. So the regenerative heart maintain a population of P1-like immature cardiac myocyte which continue proliferate and continue and contribute to the prolonged window to P28 of remuscularized injured ventricle. The pathway analysis indicate that activation of multiple factors such as PCAM2, Hippo, signaling pathways in cardiac myocyte population that are responsible for the increased activity of the machinery of the cardiomyocyte proliferation. The ongoing study aims at identifying the regulators that govern the cell cycle in large mammal. And this is important. So we have very good preliminary data that in targeting the regulators in adult heart with myocardial infarction using the modified RNA to remuscular heart with the myocardial infarction to to prevent ALV dilatation and heart failure. I will skip this. I want to thank Dr. Buxton's support for the PCTC and PCBC and PCTC and the NIH grant, and then the collaborators here that including the Minnesota colleagues, Dr. Algo, Dan Gary, Duke, Dr. Bosak, Victor Saul, Rockman in Duke, and then UAB, there's Tim Town, Rogers, Fast, and Wilcott, and Dr. Camp, Dr. and Jamie Thompson from Wisconsin Madison. Thank you.